Thanks for the opportunity to talk to you today about genome-wide association studies of COVID-19. I think I just need to fast forward through a couple of these slides of COVID-19, and in particular in relation to UK Biobank. So the motivating questions here are, why is COVID-19 a severe life-threatening disease in some but not all people? Is it because of human genetics? More generally, what's the biology of COVID-19 at the molecular and physiological level? And can we make biological discoveries that suggest existing safe drugs for new trials? A little bit of background to COVID-19. It's caused, as you know, by the coronavirus SARS-CoV-2. The virus is new to science, having emerged only in late 2019. It infects human cells by binding the ACE2 receptor on the outside of the cell. And at the severe end of the disease spectrum, it's characterized by acute pneumonia, blood clots, and heart problems. Inset, you can see, and this slide is already a few days old, there have now been confirmed over 9 million cases of COVID-19 around the world, and well over 400,000 deaths. In England, the body responsible for recording cases of COVID-19 is Public Health England. This shows you on the top the number of daily cases per day and then cumulative over the days. And what you can see is that the national epidemic in England really got started in early to mid-March, before the lockdown measures were implemented, and then they quickly curtailed the epidemic curve. And below you can see members of the UK Biobank from England, and the number of laboratory confirmed cases over the same scale. And you can see that it really tracks the national epidemic pretty well. The number of cases is around that you would expect, maybe slightly lower. In total, as of the end of the last month, there were 1,500 laboratory confirmed cases of SARS-CoV-2 infection amongst English UK Biobank participants. And I'm sorry to say that as of the end of April, there were 305 deaths with COVID-19. So there's an unusual opportunity to study the genetics of susceptibility to severe COVID-19 as the pandemic is raging around the world. One hopes that these efforts have the potential to mitigate, maybe even change the course of the epidemic. But of course, there are also challenges and perils for the scientists who are really working under the spotlight as never before. And so this is an example of a signal on the right-hand side, which came on strong towards the end of April and then quickly reversed it and is now rather modest. And I say this because really we have a challenge of statistical power. Even with 1,500 cases, that's relatively modest compared to the usual sorts of numbers of cases in human genome-wide association studies, where you might expect around 10,000 at the lower end. And so what this graph shows, not very clearly, is that at least initially we would expect to detect variants of larger effect if they exist, and also variants which are relatively common in the population. And this really is where the COVID-19 Host Genetics Initiative comes in. It's a collaboration between over 1,000 scientists around the world and 200 plus contributing studies. Broadly, two kinds of studies. There are the so-called retrospective studies, which include the large biobanks like UK Biobank, in which there are a large number of individuals who've already been studied in great detail, including they've been genotyped. And as data is linked from the likes of Public Health England, COVID-19 can be studied in those large biobanks. The second class of studies have been labeled prospective studies, and there the starting point are individuals who have severe COVID-19, and then there's an effort to study those individuals afresh and to genotype them prospectively. And you can imagine both the logistic and also the cost implications of those studies. But the opportunity is to really increase the number of cases to much more than the low thousands that we're currently looking at, because of course there's been many more cases of COVID-19 around the world. In order to bring together these disparate studies, there needs to be some harmonization of the analysis plan. And so here's a population pyramid which gives the general idea in simplified form of the 6 main analyses that have been agreed. So at the bottom you have the general population, then you have those individuals that have been laboratory tested, and here by laboratory tested I'm generally referring to the PCR-based tests for the virus nucleic acid. Those individuals that are confirmed to be infected, of those, the individuals who are hospitalized, and then at the top of the pyramid, the individuals of most concern, those with the most severe COVID-19. And on the right-hand side, you have analyses comparing each of those layers of cases to the general population. And then on the left-hand side, you have alternative analyses in which those layers of cases are compared either to other cases or other nested groups of individuals, such as all those individuals who were laboratory tested. Again, there has to be some harmonization of the analysis pipeline, and so the standardized agreed approach is to use SAGE for genome-wide association studies, which accounts for highly imbalanced case control ratios. It's a linear mixed model which accounts for population stratification. There's several groups contributing analyses of the UK Biobank to the COVID-19 Host Genetics Initiative, and in those there've been agreed inclusion criteria. So with respect to the inclusion of Public Health England confirmed cases, those individuals who were recruited to an English recruitment centre, individuals that were not lost to follow-up at the start of 2020, and individuals that don't have very close relatives also in the study. And then there are a number of post-analysis filtering steps which take into account data quality and statistical power. On May the 15th, the Host Genetics Initiative released to the public its first meta-analysis of a number of different studies. You can see the 2 UK Biobank cohorts that contributed to this initial tranche of results, those of European ancestry and those of African ancestry. The UK Biobank in this initial wave contributed the most number of cases and controls, with other large biobanks contributing strongly, such as FinGen, Lifelines, and the Netherlands Twin study. So here are the results. What I'm showing you here is for every genetic variant that was analysed, its position along the genome, and then on the y-axis, the vertical axis, the evidence of association between that genetic variant and susceptibility to COVID-19. And in particular, this analysis is looking at all those individuals who are laboratory confirmed to have SARS-CoV-2 infection versus the general population. And the blue and red horizontal lines show you the thresholds by which statistical signals are deemed to be suggestive or statistically significant after accounting for the large number of different tests. So what you can see is that there were a number of interesting signals that came up in this first wave, but there isn't really anything which is convincingly genome-wide significant right away. Another way to look at this, and I can't see this, but I can describe to you what it should say, is a so-called candidate gene approach. If one strongly suspects a particular gene might be involved, then one can study directly the effect of variation at that locus on COVID-19. And in this case, this has been published, you can see at the bottom, already in a study. It was an early, in a journal, it was an early study. And they've looked at ApoE, which confers strong susceptibility to dementia. But even after accounting for individuals with dementia in the UK Biobank, what they found was that alleles at this locus confer strong increased susceptibility to COVID-19. The p-value was around about 1 in a million, so that's consistent with the previous slide, not quite yet deemed genome-wide significant. Okay, so this slide seems to have been rotated. This is the big news in these early days of genome-wide association studies in COVID-19 is this paper, which was published in the last few days in the New England Journal of Medicine, but has been on the medRxiv preprint server for a week or two more than that. And it's a study of 2,000 cases of respiratory failure due to COVID-19 in Italy and in Spain. And respiratory failure was defined as those individuals requiring oxygen supplementation or ventilation. And although you can't see this, I can tell you that there were two signals which were genome-wide significant, one on chromosome 9 in the ABO blood group locus, and one in a cluster of genes on chromosome 3. And what was particularly convincing about this new signal on chromosome 3 is that it was independently replicated, because although the signal in chromosome 3 is not genome-wide significant in the Host Genetics Initiative, it's sufficiently strong to be deemed formally replicating in an independent study. And of course, UK Biobank was one of the larger of the biobanks contributing to that initial wave. And I think that's really what's tipped this into being very strong evidence for a host genetic contribution to COVID-19 susceptibility. Here I'm zooming in on the ABO blood group locus, which is the first of those two signals that I mentioned to you. And so what you can see is there's really only one gene involved in this signal, and that's the ABO blood group gene itself. This signal is regarded as confirmatory because, in fact, there's been a number of studies listed at the bottom which had already implicated blood group in susceptibility to COVID-19. A study in China very early on, one in New York State, and also a study which compared the frequencies of different blood groups in Chinese cases to controls comprising individuals of Chinese ancestry in the UK Biobank. And so in this Italian-Spanish cohort, what they found was that individuals with blood group A had 1.5 times higher odds of severe COVID-19, and individuals with group O blood had 1.5 times lower odds. The blood group locus encodes a glycoprotein modifying enzyme which has very far-reaching effects. Just to give you one example, plasma levels of the von Willebrand clotting factor are lowest in group O. And that might be relevant because of the unusual thrombosis traits seen in COVID disease. Another early preprint which has appeared in the last couple of weeks is starting to dig down into these results and ask what possible effect could the risk variant have. And so in this particular study, whose citation I have lost, but it's Katz et al. On medRxiv, they've asked whether the risk variant affects protein expression, and indeed it does. And they've pulled out in particular a gene called DC-SIGN. This is a cell surface expressed protein which is found on dendritic cells, part of the immune system. It's known that DC-SIGN promotes cell entry by SARS-CoV-1. And interestingly, its expression also increases with age. But it's not just this gene. The risk variant at the ABO blood group locus is involved or associated with differential expression of a large number of genes, including those involved in inflammation and cell adhesion genes. So on to the truly new signal on chromosome 3. The authors were careful to clarify that as yet the observational study, which is a GWAS, is not able to tease apart exactly what the functional variant is that's the association, but still we can dig around and see what genes are involved. And one of them is the SLC6A20 locus, which is a transporter that interacts with ACE2, the receptor by which SARS-CoV-2 gains entry to cells. CCR9 and CXCR6 are chemokine receptors that are also in this region of association, and they have functions in T-cell mediated immunity in the gut and the lung, both of which the virus is known to infect. So this Katz study has found that the risk variant is associated with differential expression of a few genes, of relatively few genes, many fewer, notably the CXCR6 ligand encoding CXCL16. And so this ligand receptor pair are implicated again in dendritic cell T-cell interactions in lipopolysaccharide-induced lung injury and inflammation, and in epithelial barrier integrity. So questions and prospects. Well, substantial increases are expected in sample size and thus statistical power through prospective genotyping studies in the COVID-19 Human Genetics Initiative. New results have been uploaded in the last week, and yesterday a new meta-analysis was performed, the results of which may be published in the next few days. So keep your eyes peeled, because there may well be new results to be found there, UK Biobank being one of the contributory studies. Improved analyses are anticipated through refinement of case definitions in UK Biobank through the imminent incorporation of hospital and then primary care records further down the line. Open questions, perhaps most topical: do genetics contribute to interracial differences in infection and mortality? There's no clear answer to that yet. What role, if any, does the HLA system and adaptive immune system play? It hasn't so far come up, but it may do. Wide open, what are the molecular mechanisms that underlie newly discovered genetic risk factors in these early GWAS? And ultimately, can these insights be exploited to improve treatment? So thanks for listening.
Now we could now press on with the Q&A. And can I start, first of all, with a question for Danny? This story that you had about von Willebrand factor and the O blood group turns out to be pretty topical in light of the impact of dexamethasone on severe mortality in COVID, because it would appear that the mineralocorticoid axis may well be the mechanism by which dexamethasone operates. And the impact of that on the feeble Palade bodies, which is where von Willebrand factor is stored, and their release is one of the suggestions of the pathophysiology of that disease. Had you thought about that as the link between levels of von Willebrand factor and the mechanism through the mineralocorticoid receptor?
Thanks for the question. That's a very technical question, and I suspect you would probably be better placed than me to answer it.
Sorry, that was a flower. That was just to keep all you guys on your toes. Anyway, I think that's an extraordinary observation you've made, and it's probably linked, actually. And it may well be that excess discharge of von Willebrand's factor may well be accounting for thrombotic complications. But it's interesting that you've come up, without knowing the plot, you've come up with a pretty interesting genetic signal that might actually tell us that that's going on. So well done for that. I
should say that it's not me personally that's responsible for that particular discovery. There were a number of papers that have implicated ABO, and the Italian-Spanish cohort is an independent study.
Yeah, right, exactly. So I think there's an interesting philosophical question about whether it's worthwhile looking for genetic markers as to whether you get infected or not, as opposed to genetic markers that affect disease progression and disease severity. I mean, there are, in HIV, I'm sorry, I've lost you. I can't hear the question. I don't know if that's me. That's probably me. Sorry.
Oh, you're back. You're back.
Can you get that? Yeah. Sorry.
Yeah, you're back. I lost you. I think you were saying there's a question about the relative role of susceptibility to infection versus subsequent severity.
Yeah, the disease progression. I guess that's really the question, because they looking around for genes that determine infection or not is actually quite a hard job. And HIV is probably the best example of where a few of those have been discovered, but there's not many of those out there in the world of infections. But I would have thought your chances were better at getting variants that actually determine extent of progression and the subphenotypes of disease, which are well known.
Indeed, yes, I would agree with all of that. Yeah, I would value your opinion on this, but it seems that perhaps the really pertinent question is, why is it that some people progress to very severe disease? We know that obviously there are important non-genetic effects, age being most prominent, but my naive supposition would be that pretty well everyone is likely to be susceptible to infection at some level. And so really the question is, why are some asymptomatic, some are mildly symptomatic, and some get severe disease?
I think that's the big question, isn't it? Is how do you get these very different phenotypes, given the fact that the vast majority of people are asymptomatic. 70% of people are asymptomatic, and yet you do get these extreme phenotypes. So I think that has to be one of the big challenging questions. Can I also ask, Danny, while I've got you, about the ApoE4 finding? A question came in from the audience asking whether you think the prevalence of dementia, which actually Gonzalo has really emphasized in his talk as well, the well-known association of ApoE4 and dementia, whether it's the fact that people with dementia are more likely to get disease in the elderly age group, because of course a lot of them care homes, they require quite a lot of hands-on management. Do you think that's the linkage to their higher susceptibility, or do you think it's something more biologically fundamental?
I think that's a great question, and I'm glad that Hilary Martin asked it. It could be, yes. So that was an analysis of all individuals that had a laboratory-confirmed SARS-CoV-2 infection versus the general population. So I agree that it could be explained by differential susceptibility to exposure, for instance.
Yeah.
But also it could be, I mean, the challenge with a candidate gene study is that by just looking at that one gene and putting blinkers onto what's going on in the surrounding genes, maybe the signal is higher in surrounding genes, or maybe there are at least other genes surrounding it which also carry the signal. So I would be happy to defer to others on the question of ApoE4. I think it's super interesting. I gather that it's also has cardiovascular effects.
It does.
So
yeah, it does.
It's an intriguing one.
No, I think it's a really interesting question. And one final question for you, Danny, again from the audience, and it was a question about the allele frequencies. Were those estimated only for the England sample or for all UK Biobank data across the laws and as well as England?
So in general, the analyses that are contributed to the Host Genetics Initiative, where they use the Public Health England laboratory confirmed cases of SARS-CoV-2 infection, one of the inclusion criteria is that the controls also are selected from English recruitment centres.
Yeah. Okay. Yeah. So you lined those up. Yeah. I think that was the basis of the question. That's great.